So hello guys and welcome back to Excelsior Academy. We'll be starting new topic, cell signaling. So basically everyone knows what is cell signaling, little bit of the basics, and then we'll move on like different types of the signaling where we'll be discussing about the tyrosine kinase and tyrosine kinase based signaling, GPCR, etc. Okay. So before that introduction, so all cells receive and respond to signals from their environment. Even the simplest bacteria can sense and swim towards the high concentration of nutrients such as glucose or amino acids. So here we know that that is known as the chemotaxis, right? So even that is a kind of your cell signaling. Many bacteria and unicellular eukaryotes, they also respond to signaling molecules secreted by the other cells. And this will help in the cell-to-cell communication. So as in the case of the biofilm formation, if you know, there's something called quorum sensing, right? So here what they do, they start secreting the molecules. What are those molecules? In the case of quorum sensing, anyone remembers? Right now I'm not getting the word. What are those molecules which is helping in biofilm formation? Yes? Anyone? I'm not able to remember right now. What are those called? Okay, maybe I can get it from the, in the next lecture, I'll get it for you. So, so the thing is that they secrete certain kinds of signals, okay, which will help, which will give the signal to the other bacteria to come together. And then they start coming together and then they start forming a small colony and then start secreting all those extracellular lipopolysaccharides and they form a sheet-like structure, okay, glycocalyx they form on the top of it, and then they initiate the biofilm formation. So as you know, mainly Vibrio, Pseudomonas, all these bacteria are responsible for formation of biofilm. Yeah, so those words are, so that word is autoinducers. So they secrete out autoinducers, that is, they themselves give the signal to them and to the nearby cells. So these autoinducers, then they are going to get all the signals coming together, making sure that all the bacteria coming together and start forming the biofilm. Okay, so that is also a part of your cell-cell communication. Mating between E cells, for example, is signaled by peptides that are secreted by one cell and bind to the receptors on the surface of the another. So this is also a kind of cell signaling. Now, signaling molecules and the receptors. So different kinds are available. They varies in size, structure, and polarity. Structurally, the signaling molecules used by plants and animals range in complexity from simple gases to proteins. Now, if I say that nitric oxide, it's a gas, right? Do you think that this is also responsible or this can act as a signal molecule? Krishita, do you think that nitric oxide can act as a signal molecule?
Yes, ma'am.
Yes, it is. And we'll see how even the NO-based signal molecules are helping in the vasodilation. Okay. So it acts as a signal molecule over there and results in the activation of the receptors, which will result in the vasodilations of the blood vessels. Okay. Now, some of these molecules carry signals over long distance, whereas others are locally to convey information between the neighboring cells. So mainly the molecules which carry signals over long distance in the case of the humans are your hormones. Okay. Now, if it is between neighboring cells, so in the case of the neurons we have seen where they secrete our neurotransmitters, right? So in the case of postsynaptic and presynaptic neuron, we have seen if this is a synapse, let's say this is a synapse. So this is junction between two cells, neighboring cells. So this is your presynaptic neuron, this is your postsynaptic neuron. So here we have seen the molecules, for example, in the case of the nerve impulse, acetylcholine is getting released, right? And then they go and bind to the receptors which are present over here. So then this is also a kind of your cell-to-cell signaling, correct? Now, there are three types of signaling involved. Okay, we'll come to that later. In addition, signaling molecules differ in their mode of action on the target cells. Some are able to cross the plasma membrane, where some are able to bind to the intracellular receptors to the cytoplasm or nucleus, where others are going to bind on the cell surface receptor itself. Okay. Now coming to the modes of cell to cell signaling. So there are three types are there. I hope everyone knows also that autocrine, paracrine, and last one is anyone? Autocrine, paracrine, and third one is Tanvi. Any idea? It is your endocrine. Okay. So cell signaling, either from the direct interaction of a cell with its neighbor or from the action of the secreted signaling molecule. So there are two types are there. So basically in the case of cell-cell signaling, that is direct cell-to-cell signaling, as we saw in the case of the leukocytes and endothelial cells. So their selectins are binding to the carbohydrate moiety present on the receptors of the endothelial cells. ठीक है? एक वो हो गया आपका direct cell-to-cell signaling, like where the receptors and the ligands are present on the surface of the two cells and then they are interacting. दूसरा मेथड क्या हो गया? Either one of the cell is gonna secrete out the chemicals, okay, and then that chemical is gonna bind to the receptor. So the chemical is gonna act as a ligand and it is going to bind to the receptor. So these are the two ways by which a cell signaling is carried out. Now signaling via such direct cell-to-cell interactions plays critical role in regulating the many interactions between different types of cells that takes place during embryonic development as well as in the maintenance of the adult tissues. Okay. Now coming to the modes of signaling, the third one which I was talking about, the endocrine signaling. So endocrine signaling, here mainly they talk about the hormones which are, you know, for the long distance. So they go for the long distance. Now why long distance? Because they need to be carried out through they need to be carried through the circulation. Okay, so they get into the blood system and with the help of the circulatory system they reach their target cell. Okay, now signaling molecules like hormones are secreted by specialized endocrine cells. They are carried through the circulation to act on the target cells which are present at the distant body sites. For example, steroid hormone estrogen, which is produced by the ovaries and stimulate the development and maintenance of the female reproductive system and the secondary sex characteristics. Okay, so what is happening from the endocrine or the, you know, the ovaries, these hormones are being released. They get into the circulatory system and then they reach the target cell. Okay, now what are the secondary sex characteristics of a female? So when the estrogen or these hormones are released, that start developing the breast or then whatever, then those the other secondary sex characteristics, whatever is present. Okay, now we'll talk about this in detail, like how the hormones are actually getting released and how they are being traveled through the circulatory system and reaching the target cell. So there are many examples like how these signaling is being carried out. We'll discuss later. Now the second is the paracrine signaling, a molecule which is released by one cell. It acts on the neighboring target cell. So ये क्या हो गया? जैसे कि in the case of our neurons we have seen that in the example of the neurotransmitters, right? So from one cell it is being released and it goes and binds to the receptor present on the neighboring cells. Now in autocrine क्या है? You self-activate. So mainly in the cancer cells this is happening. Okay, the immune cells तो करते ही हैं, but the main a very good example can be given by the cancer cells. So in the case of the autocrine signaling, some cells respond to single molecules that they themselves produce. So that is self-signaling. आप ही ligands produce करोगे and it will go and come and bind to your own self receptors. Okay, so the response of these cells of the vertebrate immune system to the foreign antigen. Certain types of T lymphocytes respond to antigenic stimulation by synthesizing a growth factor that drives their own proliferation, thereby increasing the number of the T lymphocytes. Okay, and that's how we get our immune response. Now, another example is where cancer cell is going to produce a growth factor, which itself is going to respond. Okay, and that's why they are continuously driving its own unregulated proliferation. See, अगर कैसा होता है, इसको अगर किसी signal की जरूरत है in order to proliferate, and that signal needs to come from some other source, then the cancer cells can be regulated, right? The spread or the unregulated proliferation of this cancer cell can be regulated. But since it is not depending on the other cells, It is like, its own producing its VEGF or FGF or the other EGFR. So all these factors, they themselves produce, the chemicals goes out, they go and bind to the self receptor. Now, here one doubt should come to your mind. Suppose let's say that this is a cancer cell. It is producing the VEGF factor. Okay. Now VEGF factor is for your vascular endothelial growth factor. That is, it is helping in the angiogenesis. What is angiogenesis? Formation of your new blood vessels. Okay. Formation of new blood vessels. Now, what question should come over here? Okay, right now we said that in the case of the autocrine signaling, the VEGF which is produced, they can go and bind to itself. Okay, so in the receptor, so it goes and binds. Okay, now this VEGF, now why they go and bind? So that they can secrete more VEGF. The short meaning of autocrine signaling is that they themselves can bind and help in more production. Now, can this VEGF can bind to the another cancer cell in order to proliferate or help that another cancer cell also to proliferate? Is it possible? Akshaya, is my question clear to you?
Yes, ma'am.
Okay, so what do you think then?
If the signal is autocrine, then it cannot activate the neighbor cells.
Okay, but this is present, right? Let us say that it is also present. This cell is also present in the nearby. In short, the question is, can this molecule act for both autocrine and paracrine signaling? Prashita, what are your thoughts?
I feel yes, it should, because angiogenesis, there is this vascular development which takes place between the cells. So I think it should work, but
yes, it will, right? See, VEGF, when it is secreted out, it is not saying, or it is not having that clear-cut tag that this is secreted by this particular cell only. It is present in the environment. So this particular cancer cell can also take. Okay. So when the cells are present in the nearby, okay, this same chemical molecule can act as autocrine signaling molecule. It can also act as paracrine signaling molecule. Okay, so there is no clear-cut distinction saying that when one particular cancer cell is producing, it should bind to that only. No, that chemical is irrespective of its origin. Okay, so if the receptor is present on both the cells or any other nearby cell, definitely it can bind. Okay, now it's clear?
yes, ma'am.
Yeah. So the example of steroid hormones and the nuclear receptor superfamily. So here we are taking one example of endocrine signaling. Okay. So here in this case, in the steroid hormones, so it is going to use intracellular proteins for this signaling. Okay. Now why intracellular? Because to any cell, as we already mentioned, steroid hormones means it is going to be your endocrine signaling, correct? Now, endocrine signaling means it is going to be secreted from one cell and it enters your circulatory system and it is going to enter the target cell, right? Now, it is going to enter the target cell. So, steroid, they are hydrophobic in nature, correct? So, they are hydrophobic in nature. So, they can enter the cell directly, passing through the plasma membrane. Agree? So, they can enter into the plasma membrane of the target cell and also to your nuclear envelope. Agree?
Yes, ma'am.
Yeah. So, what will happen then? Now, these steroid hormones can act at the expression level. So these steroid hormones can act as the affecting the transcription level of your gene. Okay. And then either to, regulate or to inhibit or to enhance the production of that particular gene. Okay. So same thing we are going to see now. So it uses intracellular proteins for the signaling. Why? Because it enters into the cell in the cytoplasm and then from the cytoplasm to the nucleus. So that's why in order to take steroid hormones inside, you need intracellular proteins. If you remember, when we were talking about the transport of the proteins into the nucleus and out, importin, exportin, right? So those proteins or those receptors are required to bind or to recognize the steroid hormones and to take them inside of the nucleus. Okay, so those are those intracellular proteins. Now, these intracellular receptors respond to small hydrophobic signaling molecules which are able to diffuse across the plasma membrane. For example, steroid hormones, vitamin D3, retinoic acid, etc. The others are testosterone, estrogen, progesterone, and the corticosteroids. Everything which is synthesized from the cholesterol. Now, testosterone, estrogen, progesterone, what are these? These are the sex steroids produced by the gonads. Corticosteroids, including glucocorticoids, are produced by the adrenal glands. So they act on the variety of cells to stimulate the production of the glucose. Okay, so these are the example of your steroid hormones. Now, these structures, as you can see, all these are having very similar structure to your cholesterol. And then we have got the thyroid hormone T3, T4, vitamin D3, retinoic acids. So what do we see from this structure? What do we understand? That they are highly hydrophobic in nature, right? Now, thyroid hormone is synthesized from the tyrosine in the thyroid gland. Now, guys, this is also important, though we will be covering this in the biochemistry, but if it is possible, study this, like the synthesis. Okay, the synthesis of thyroid hormone from tyrosine in the thyroid gland. It plays important roles in the development and regulation of the metabolism. Now, how tyrosine in the thyroid gland and what are the enzymes required for the synthesis of thyroid hormone? That is important. Okay, so anyone, Krishita or Akshaya, Tanvi, do you have any idea like how T3 and T4 are synthesized? Thoda sa bhi idea? Yes, anyone? We have done this like in your biochemistry, it is there. Yes, कोई idea है? Okay, no issue. What you can do is just try to go through it, okay, and try to get the answer. Like what are the intermediate or what are the, the enzymes involved in this? Okay. It's a small assignment that you can go through it. If you're not getting it, then let me know. Okay, I'll share the pathway with you guys, like how thyroid hormone is synthesized. Okay, so this is small assignment for you. Get the thyroid hormone synthesis, okay, T3 and T4. Now, vitamin D3 regulates calcium, 2+ metabolism and bone growth. So we know the relation between the vitamin D and calcium. Retinoic acid and related compounds synthesized from vitamin A play important roles in the vertebrate development. So this also we know. कहीं पे retinoic acid होगा, retinal होगा, so that is nothing but your vitamin A, okay, and helps in the vision. Okay. Now, if anyone is asking you like how retinol or retinal is, there is a difference, okay? Retinol, retinoic acid and retinal. When retinol is said, it is your alcoholic group present. Retinoic acid, carboxyl group is present. Retinal means anyone? Akshaya, retinal means which group is present?
Aldehyde.
Yes, very good. Aldehyde is present. Okay. So I have taken this in one of the previous lecture. How retinal is helping in the vision. So when light falls on roads and cones, what happens to the retinal, or like which receptor is involved?
Here the phototransduction comes.
Yes, yes. So what happens when light is falling on the retinal? How do you understand that, light has fallen and how your brain is able to recognize, like making the image what is seen through your eyes?
That is converted into cis, and there is this pathway cascade which follows it.
Yes, you are going on the correct path. So what is that receptor on which retinal is present, and when light falls on it, what is the name of the receptor?
Rots.
What is it?
GPCR?
By nature, they are GPCR, but what is the name of it? Light sensitive receptor. एक और नाम है इसका.
Rhodopsin?
The
yes,
rods, no?
Yes, rhodopsin. It is rhodopsin onto which your light falls, right? And then the phototransaction takes place, wherein which the retinal cis and trans conversion is taking place. Okay. Rods and cones तो cells हो गए जिसके अंदर rhodopsin है. Okay. Okay. Now, because of their hydrophobic character, the steroid hormones are able to enter the plasma membrane, right? The passage of thyroid hormones across the plasma membrane then again facilitated by carrier proteins. As I said, it needs to be bound to the protein so that they can be taken inside of the nucleus. Okay. So once they're inside the cell, these steroids like thyroid hormone, vitamin D3, retinoic acid, they will bind to the intracellular receptors expressed by the hormonally responsive target cells. Now, these receptors, they are nothing but the family of proteins known as the nuclear receptor superfamily. Okay, so they are the nuclear receptor superfamily. They are the transcription factor that contain related domains for ligand binding, DNA binding, and transcriptional activation. So when they bind, they get either activated or repressed, and accordingly, they regulate the gene expression. Okay, so this already mentioned. Let me see if I have the figure. Yes. Okay. So here the action is given of glucocorticoid action. Okay. So again, as I said, it is hydrophobic in nature. Okay. Anyone would like to try giving the answer? Krishita, can you explain the figure? Just try once from your side.
I just need to go through it once.
Yes, you can read the description given over there so that you get an idea.
Okay, so there is this glucocorticoids, which would, initially the receptors are bound to HSP90. That is, they are in an inactivated form, but as soon as the glucocorticoids comes in, when it passes traverses through the cell, that's when this glucocorticoids is going to bind to them, and these receptors, they are going to dimerize. And once the dimerization happens, they would be able to get into the nucleus. And once they get into the nucleus, they associate with the, they bind to the DNA and they associate with the coactivator they associate with the coactivator with the histone acetyl transferase activity to stimulate. Then there would be a stimulation of the activity. Yeah, that is it. I couldn't read because the last sentence is not visible on my screen now.
Okay, but so this is correct. So what is happening? Any steroid molecule, okay, glucocorticoid or any other steroid hormones, when it is present, you have got the hydrophobic nature, right? So they can easily diffuse through the plasma membrane. So when I say easily diffuse, it means no receptor is required here. Okay, so they can just pass through the plasma membrane of that particular hormone responsive cell. Now, when they go, they bind to their inactive glucocorticoid receptor, which is bound to the HSP 90. Now, why again 90? So that this particular, which is in inactive state, does not lose its structure, does not lose its function. That's why it is bound to the HSP 90, right? Now, once they go and bind to the receptor, glucocorticoid receptor, HSP 90 leaves, they get dimerized. That is when they get activated. Good. Then it enters the nucleus. Now what is happening? It goes and binds to that particular gene region which needs to be regulated. Okay. So here as such in this group, it is not shown, but what they have shown is that now if suppose this is the region for the gene expression, the transcription should take place, then of course the histones need to be removed so that the DNA get free. Agree? Now, how it can be removed? When the histone acetyltransferase is there, right? So what they can do, they will add the acetyl group to these histones molecules. What will happen then? Just because of the DNA ka negative charge and histones ka positive charge, they are in interacting way, right? Now, once you add acetyl group, which is again a negative charge, what will happen then?
The histones would interact with acetyl group instead.
Yes, and because of this negative interaction, what will happen? The histones will leave the DNA because negative charge is going to repel each other, right? So histones will get removed. Now, once they remove, the DNA is free, gene expression will take place, your RNA polymerase will come, it will transcribe the mRNA, etc., will take place. Okay, so that's how these steroid or the glucocorticoids are giving the instructions to the gene so that they can continue for the transcription. Okay, so this is how the glucocorticoid is going to act. Okay, now what is the function of glucocorticoid? Glucocorticoid. Yes, so what is the function of this particular molecule?
Ma'am, to stimulate glucose secretion.
Glucose secretion?
Corticoid.
So when you say glucose secretion, secretion kahan se? See, do you know something called ACTH?
Yes, adrenocorticotrophin hormone.
Yes, right. It is an adrenocorticotrophic hormone. Iska kya kaam hai? So when there is this flight or fight response, okay, this is the time when you need maximum of your glucose, चाहे भागने के लिए या डर के लिए. Adrenal cortex, adrenal gland से it is being released. Correct. So they are released from the adrenal glands. Yani ki it's a endocrine signal response, right? So what happens? This results in the activation of your ACTH response, which in results need more amount of glucose. Now, how can you release more amount of glucose in the cell? There are two ways by which you can get maximum concentration of glucose. What are those two ways? Yes.
Glucogenolysis.
Glucogenolysis कैसा शब्द है? Either you have glycogenolysis.
Sorry, glycogenolysis.
Or you have glycolysis. Okay. So yes, this is one of the method by which you can have more concentration of glucose. Then what is the another way? Glycolysis means using up of the glucose, but that time I need glucose. Correct?
Yes, so glycogenolysis.
Or दूसरा? So this is one method in second. What is another way by which you can get the glucose?
Fat breaking down, no, not glucose, the energy basically that acetyl CoA का.
नहीं इतना टाइम नहीं है. जब मेरे ऊपर मौत खड़ी है सामने, my body doesn't have time to break down the fat that time. It will take long procedure. Shortcut चाहिए. I need turant, you know, the sources of energy. If you know the first stored form of energy, that is glycogen, सबसे पहले glucose आपको glycogen से ही आएगा, right? When I need एकदम immediately. Then comes from the adipose tissues, जो fat and all. What is the another way? If I need it immediately. सोचो, the answer is there itself. और क्या कर सकते हैं जिससे ग्लूकोज बढ़े बॉडी में? अच्छा,
Is it through consumption?
Krishna, think about a situation, right, where you are scared and you are running. Are you able to eat that time? नहीं ना?
Increase glucagon.
Huh?
Glucagon.
Okay, glucagon is doing what? Again, that is coming at that level, hormone level. I'm asking what else can be done? The pathway. So all this glucagon, glucocorticoids, ये सब एक दूसरे को activate करते so that at the end glycogenolysis is happening. And one more. What is this? Okay, I'll give you the hint. कोई चीज अगर आप तोड़ के बना सकते हो तो कोई चीज जोड़ के भी बना सकते हो. Now what is the answer? So this is something you're breaking and making glucose. What if you can't create or synthesize glucose?
Amino acids?
Yes, and it results into what? What is that pathway? Arey Baba, it is gluconeogenesis. Right? Don't tell me you have not heard of it.
Yes, we have heard.
Okay, so these are the two ways by which you can get high concentration of glucose at the flight fight response. Okay, yeah. Moving on to the next. Yes. Okay, so now what if that hormone is not present? Okay, before that, Priscilla, do you want to discuss something else?
Yes, ma'am. I just wanted to know that gluconeogenesis, is it a much faster process? Like I know fat would take a lot of time because they are present in a reduced form.
Yes.
But then first preference would be glycogenolysis and then gluconeogenesis. Right?
Yes, yes. Now, why so? Because see, in order to carry out your gluconeogenesis, if you know glycolysis enzymes are like, you know, constitutively expressed, right?
Yes.
Yes. And if you see the difference between the glycolysis and gluconeogenesis, if you remember, there are only 3 steps which is different. Rest all is same. So
it's a reverse.
Yeah. So it becomes easy reversible step for the gluconeogenesis, because already see all the cells always glycolysis is happening. It just that all of a sudden certain situation is coming where you need high amount of glucose. So that time obviously the glycolysis will stop. Okay, of course the glucagon and insulin will be acting, but that time when you need gluconeogenesis, it's just a matter of 3 enzymes rather than, you know, breaking your fat. Because see, I know fat gives the maximum amount of energy, but the breakdown is taking so much amount of energy there. And when you get the acetyl-CoA, you know, lot of fat burning process means what? You need so much of oxygen also. But when you're running, when you're in that state, if you know, we do the oxygen supply to all the cells is not proper, right? That time only thing is that, and the glucose which is taken, see at the end we need glucose, right? And at that time when you are running and also oxygen supply is also not adequate. So what happens? This glucose goes for your lactic acid accumulation. That's why you get all those pain and all when you suddenly run and sit, then, you know. So that time, just to be out of the danger situation, we don't go with the normal glycolysis. We go for the lactic acid accumulation. And later when you are in normal state where oxygen supply is proper, then you get all those response. Okay.
Okay.
Yeah. Now, coming back over here. So if gene regulation is happening because of the thyroid hormone receptor, okay. So there what we saw, that glucocorticoid is coming, binding to its receptor, going to the nucleus. What if there is no hormone? What if there is no hormone? So what will happen? So in the case of the absence of the hormones, instead of HAT, you will have HDAC. Okay, what is HDAC? It is your histone deacetylase activity. So it will make sure that the histones are not having any acetyl group. So they will be bound to your DNA, right? So nucleosomes assembly will be present. And this thyroid hormone receptor, they will be without any hormones. Then all of a sudden, if you add a hormone, then the transcription will get activated. Otherwise, there is no gene expression. Clear? This part is clear? Simple, like what we just discussed now, right? So here, when we were talking about the hormones are present and HAT gets activated. When hormone is not present, instead of HAT, you have got the HDAC. So in the presence of hormones, transcription is happening. In the absence of hormone, transcription is not happening. Simple? Clear?
Yes, ma'am.
Yeah.
Yes.
Now coming to the signaling by the other small molecules, right? So as just beginning of the lecture, when I asked like, can nitric oxide can act as a signaling molecule or not? Yes, it can. Acetylcholine, GABA, and the auxin. So all these are nothing but acting as a signals for each of your cell in order to respond. Okay. Now, in this lecture today, let's discuss about the NO. Now, it's a simple gas, nitric oxide is a major paracrine signaling molecule in the nervous system, immune system, and the circulatory system. It is able to diffuse directly across the plasma membrane of its target cell. Now, why does NO does not need any receptor? Because it's small in size.
Gas.
Yes, Krishita, you were saying something?
It's a gas, no?
It's a gas and small in size, so it can easily pass through the plasma membrane. Now, in the molecular basis of NO action, it binds to a receptor that regulates transcription. It alters the activity of intracellular target enzyme called guanyl cyclase. Now, what does guanyl cyclase does? It converts your GTP to your cGMP, that is cyclic GMP. For example, dilations of blood vessels. Okay. We do have adenylyl cyclase also similar to this, which results in the synthesis of cyclic AMP. Similar to that is guanyl cyclase. Now, dilations of blood vessels, that is the nitric oxide action. Now, the first step in the process is the release of neurotransmitters such as acetylcholine from the termini of nerve cells in the blood vessel wall. Okay, let's go slowly because this is really very important to understand. It's a part of your animal physiology also. So, what is happening? First, the release of the neurotransmitters. Now, these neurotransmitters will act on the endothelial cells to stimulate NO synthesis. Okay, so we are talking about the blood vessels. Blood vessels are made up of endothelial cells. So, what had happened? So, neuron blood vessels का जो रहता है आपका connections, there the signal has been received. Neurotransmitters acetylcholine has been released, okay, which is giving you the signal to stimulate the NO synthesis. Now it diffuses to the neighboring smooth muscles where it activates guanyl cyclase, resulting in increased concentration of cyclic GMP, which will induce muscle cell relaxation and blood vessel dilation. अब जब muscle cell relax होगा, then only the dilation can happen. Okay, in the stress condition, it cannot. Now, it is also responsible for signaling the dilation of blood vessels that leads to penile erection. So, when muscles is relaxed, it dilates. Okay, it is also interesting to note that the medical use of nitroglycerin in treatment of heart disease is also based on its conversion to NO, which dilates blood vessels and increases the blood flow to the heart. So in this case, what is happening in most of the heart disease, as you know, the blood vessels are congested. Okay, कैसा होता है, cholesterol बहुत ऐसे settle down हो जाते हैं, right? So because of which it becomes very narrow for narrow for your blood to pass through the arteries, and because of which what will happen, it's a pressure on the heart, right, to pump with so much of pressure through those narrow vessels. So in order to treat that particular heart condition, they add nitroglycerin. So what will happen with the nitroglycerin? These blood vessels, they will dilate and increases the diameter so that the blood can flow easily. Okay, so this is one example of the treatment. Now let's watch this video where you can see the action of NO.
Nitric oxide and vasodilation. Binding of acetylcholine to its vascular endothelial receptors triggers a cascade of reactions that result in excessive release of calcium ions from their indigenous storage sites. Calcium ions bind to their messenger protein, calmodulin. The calcium calmodulin complex activates the nitric synthase enzyme. The active form of this enzyme catalyzes the conversion of arginine amino acid and oxygen into citrulline and nitric oxide molecule. To exert its vasodilatory effects, the newly produced nitric oxide molecules diffuse into the nearby smooth muscle cells. In the smooth muscle cells, nitric oxide triggers a cascade of reactions, starting with activation of a guanyl cyclase enzyme. Active guanyl cyclase in turn activates the conversion of GTP into its active form, cyclic GMP. Cyclic GMP facilitates the dephosphorylation of myosin light chains. This dephosphorylation process induces the dissociation of myosin and actin filaments and results in smooth muscle relaxation. The pharmacological nitric oxide donors, such as organic nitrates, pass through smooth muscle cells and release their nitric oxide into their cytoplasm. The external nitric oxide mimics the physiological functions of indigenous nitric oxide, which usually end by smooth muscle relaxation. Some nitric oxide donors, such as sodium nitroprusside, release their nitric oxide in the blood. Then, nitric oxide molecules diffuse into the smooth muscle cells, where they cause them to relax. Nitric oxide donors include sodium nitroprusside and organic nitrates such as glycerin trinitrates, isosorbide dinitrates, and isosorbide mononitrates.
Okay. So guys, did you get it? Like how NO is actually responding? Okay. Is it clear?
Yes.
Okay. So the release of neurotransmitters is signaled by the arrival of an action potential at the terminus of the neuron. So we just started with the acetylcholine release, right? Now before that, as you know, whenever action potential is there is exchange of the ions, and that's how there is a signal that acetylcholine should be synthesized, and in the vesicle they should go to the synapse where it's going to get released. So this already we have done it, right? So the neurotransmitter then diffuse across the synaptic cleft to act on a target cell. since they're hydrophilic, they're unable to cross the target cell. So they will go and bind to its receptor. So ligand-gated channels, which we have already discussed in the case of the acetylcholine. Now, neurotransmitter will bind to these receptor, induces a conformational change, which will open the ion channels, which will result in the ion flux in the target cell. So that's how all these, you get a signal, okay? Neurotransmitters are released. And then this gives a signal to the others, like how the NO is being secreted out. And then it gives the, at the end, it gives the signal for the vasodilation. Okay, so this is like long process. Now coming to the peptide hormones and the growth factors. So the widest variety of signaling molecules in animals are the peptides, ranging in size from only few to 100 amino acids. It includes examples like neuropeptides, peptide hormones, and a diverse array of polypeptide growth factors. So let's talk an example of insulin. So this is, and the hormones which are produced by the pituitary gland, like growth hormone, FSH, prolactin, and the others. Neuropeptide, it includes enkephalins and endorphins, acting as natural analgesics that decrease the pain response in the CNS. Okay, so see guys, see, this is something which you need to remember and understand. So enkephalins and the endorphins are natural analgesics. So like we take paracetamols or Combiflam. So what are those? They are also analgesics, right? If you have certain pain, headache ho raha hai, to aap kya karte ho? We take these medicines, right? The other way is, the natural way is in which case, like as I say, it is also present in your choco. Okay, so that's why many a times you must have seen people whenever they're in pain or they are depressed, okay, they feel stressed out. So what happens is that when your brain is not able to release such neuropeptides, you seek for them from the outside source. So you can see many, I mean, obviously many girls get easily depressed, so they go for the chocolate thing. Okay, so in the dark chocolate or the chocolate, you can have all these neuropeptides which will help in, decreasing the pain response in your CNS. And the other case is like where you can have potatoes. So you can see people eating chips also a lot. So they are discovered during the study of drug addiction. They are naturally occurring compounds that bind to the same receptors on the surface of brain cells as morphine does. Okay, so that's why sometimes I feel, why can't they just go for the chocolate and, potatoes instead of going for morphine? Now, can anyone tell me why still people are, getting addicted to morphine, but not to the chocolate and potatoes, whereas it is the same molecule which goes in binds and give the pain relieving response? Any idea? But still people are getting addicted to morphine.
May vary.
Yes.
Concentration may vary.
Then you have not so much of chocolate. Chocolate is more cheaper. Potato is more cheaper than morphine. If you say that this 1 microgram is more effective than 1 milligram of the chocolate wala, then eat this more. It is something else. What is it? You're in the right path, Krishita, but so what it can be? It is not the concentration, but it is something else.
The hormone level may be less when compared to, maybe morphine releases a lot of hormones which we might get addicted to.
Akshay, not clear, not clear. Can you repeat? I mean, voice was not clear.
So the chocolates and potatoes, they have the serotonin split. So the amount of hormones released will be comparatively less when compared to morphine.
Okay, correct. But there is also one more point to this. Okay, I'll give you the hint. Tanvi, I'm expecting this answer from you, okay? So as Akshay said, it is the amount of the hormones getting activated, right? By the chocolate and the morphine is different. It is also something about the effect. एक morphine लेने के बाद how the person is behaving and the chocolate and potato.
Is it related to metabolism? Like here, it directly affects the metabolism and that would take some while.
Yes, but उससे भी एक से पहले एक और है. There's something about that. Yes, that is true. So the metabolism of chocolate-related components will be fast as compared to the morphine, and because of which the effect of morphine lasts for long, as compared to the chocolate and potatoes. What is that thing called? It is the affinity. Okay, it is the affinity by which it binds to its receptor. So it is very strong, as in the case of this morphine. इसी वजह से if you take only 1 microgram of this content as compared to the 1 milligram of the chocolate components, it is going to have huge effect. Okay, so if you see, you must have seen people taking drugs in movies and all. They, ऐसे छोटे packet में ही आता है, and they take hardly of this much small amount they take. Either they inhale it or they put it or they inject it, right? So the concentration is very less, but when it goes and binds to the receptor, the affinity is very strong. And that's why the metabolism of this drug is taking, it's very late. And the effect is for long. यानी कि अगर आज आपने लिया है, so it will give you that nasha for the another 1 or 2 days for a normal person. Okay, but whereas chocolate is for that particular time till you're eating, and that's why we then we tend to eat more chocolate, or we tend to eat more of the junk. Okay, because the response time is less for this particular thing as compared to the morphine. Okay, good. So everyone had an idea, right, how it is working. Okay, so let's take one example, that is epidermal growth factor, which stimulates the cell proliferation. So it is a 53 amino acid polypeptide. It has served as the prototype of a large array of growth factors, which play important roles in controlling the animal cell proliferation, both at the embryonic level as well as at the adult organisms. Then we have got the platelet-derived growth factor involved in the wound healing. So it is stored in the blood platelets released during the blood injury at the site of the wound. It then stimulates the proliferation and movement of the fibroblasts in the vicinity of the clot, thereby contributing to regrowth of the damaged tissue. See guys, now you can see like how everything is getting connected. So we have seen how fibroblast moves, right? The movement of the fibroblasts. We have talked about the actin filaments. We have talked about the adhesion or the focal adhesion which is forming between the fibroblast and the surface, like how it is making with the extracellular matrix so that it can start moving. Right now, this all proliferation, the movement of the fibroblast is all because of the PDGF. Now, PDGF is released, so it all results in this particular movement of the fibroblast. PDGF kahan se release hoga? From the blood platelets. Now, why it is releasing? Because there is a wound. Okay, so we need these fibroblasts to come together and will help in the clot formation. So when there is a tissue injury, this gets activated and then coagulation cascade starts. So of course the PDGF is released. Along with that, the EGF is also released, VEGF factor is also released, and then the chemotaxis of the inflammation cells, mitogenesis, all this will start so that the wound can be sealed off or the clot can be formed. Okay, now coming to the G proteins and the cyclic AMP. Okay, let's continue then. Everyone okay with it?
Yes, ma'am.
Okay. So coming to the G proteins and the cyclic AMP. So most ligands which are responsible for cell signaling, including the neurotransmitters, peptide hormones, growth factor, they all bind to the receptors on the surface of their target cells. The cell surface receptor, including the receptors for the peptide hormones and growth factors, are regulated by the activity of the intracellular target enzyme. Just now we saw, right? Now these enzymes then transmit signals from the receptor to the other intracellular targets, which results in the signal transaction. So see, that's why I'm saying do not get confused when we say cell signaling is different from your signal transaction. Okay, and what is the difference over here? Cell signaling matlab cell ko bahar se signal mila hai from the environment or from the neighboring cell. So that particular ligand will go and bind to the receptor present on the cell surface. This is cell signaling, or this particular cell is giving or releasing something else for the environment. That is cell signaling. What is signal transaction? After getting the signal from here, okay, the ligand comes and binds with this receptor. Yahan se leke till gene expression, what is happening? based on which the cell is gonna behave. Okay, either it is going to move or it is going for the apoptosis or whatever it is. So these enzymes which are present downstreams are getting activated, or the intracellular targets. Okay, so this cascade is known as the signal transduction. Clear? Now, the targets of such signaling pathways frequently include transcription factors, which is going to help in the gene regulation. Ligand binding to receptor on the surface of the cell initiates the chain of intracellular reactions, reaching the target cell nucleus and altering the programs of the gene expression. Same thing, which I just explained now here. So GPCR and the G protein. So as you can see, GPCR, they are made up of 7 alpha helical transmembrane proteins. Okay, what you have to remember here is that it is a 7 alpha helical proteins. N terminal is outside of the cell and C terminal is present in the cytoplasm. This is important. It is the largest family of cell surface receptor which transmits signals to the intracellular targets via the intermediate actions of the guanine nucleotide binding proteins called the G proteins. Okay, so they are also responsible for the smell, sight, and taste. All your olfactory receptors, they are nothing but the GPCR. Sight, just now we discussed rhodopsin. Smell, everything is based on your GPCR. Okay, so the G protein coupled receptors, they are structurally and functionally related proteins characterized by 7 membrane spanning alpha helices. Please do remember, okay, important hai ye. Okay, so the binding of the ligands to the extracellular domain of this particular receptor results in the conformational change that allows your cytosolic domain receptor to activate the G protein. Okay, so what is happening outside of a cell, the hormone is there, it goes and binds to the GPCR. Now, GPCR, it has got this cytosolic domain. So there's a conformational change. When this conformational change is there, it is going to activate your G protein, which is composed of your alpha, beta, and gamma. Okay, now then depending on the signal, whether it is stimulatory or inhibitory, the G protein will get activated. So if it is stimulatory, alpha will get activated. If it is inhibitory, beta, gamma will get activated. Okay, so the activated G protein then dissociates from the receptor, carries the signal to the intracellular target, which may be either an enzyme or an ion channel. So depending on that, it will get activated. So hormone is there, goes and binds to the receptor. The cytosolic domain is going to activate your G protein. So this binding induces a conformational change in the GPCR, sorry, conformational change in the GPCR results in the activation of your G protein. So which is made up of, it's a trimeric protein, alpha, beta, gamma is there. Beta, gamma is always together, alpha can get dissociated. Now, if it is a stimulatory, then alpha will get activated, which will go and activate your adenylate cyclase, which in turn will result in the increased concentration of your cyclic AMP. Clear, guys? Is it clear? This path is clear? Yes, everyone. Akshaya, would you like to repeat once again?
Oh, okay, ma'am. So where there is a signal, the signal is a hormone when it comes and binds to a receptor. So when the signal is inhibitory, the beta and gamma subunits get activated. Beta and gamma subunits are always together. And when it is an stimulatory signal, so the G alpha gets stimulated, and this activated G alpha goes and binds to the adenylate cyclase, and adenylate cyclase helps to form the CAMP.
Yes. Okay, good. Okay, now coming to the structure, I mean, I already mentioned though. So it consists of 3 subunits designated as alpha, beta, and gamma. They are frequently called heterotrimeric G proteins to distinguish them from the other guanine nucleotide binding proteins like RAS and RAS related proteins, which are involved in the nuclear import RAN, vesicular transport, ARF and TRAB. All this we have discussed, guys, right? So this is your GPCR, 7 alpha helices binding cytosolic domain. Then we have got over here alpha, beta, gamma associated. This together, everything is known as your GPCR. Now, regulation of G proteins, again, already discussed. So the alpha subunit of the heterotrimeric G proteins binds guanine nucleotides which regulate the G protein activity. In resting state, alpha is bound to beta and gamma along with the GDP. Hormone binding induces conformational change in the receptor, such as the cytosolic domain of the receptor, which interacts with the G protein. Now, the activated receptor then acts as guanine nucleotide exchange factor to stimulate the release of GDP to the GTP. So this also we know, like many times we have seen also, right? So you have got the guanine exchange factor, which will release GDP and GTP will come. So the activated GTP bound alpha then dissociates from beta and gamma. So when the signal is stimulatory, okay, then only the alpha getting activated means GDP ke jagah GTP aayega. So the activity of alpha subunit is terminated by the hydrolysis of the bound GTP, which is stimulated by RGS, that is regulator of G protein signaling, which act as GTPase activating protein gap for the heterotrimeric G protein. Now the inactive alpha subunit, again it will go back to the beta gamma and it is ready for the another round of cycle for the signal. Okay. So in human, we have got 21 different alpha, 6 beta, 12 gamma, leave it, not important. Different G protein associates with different receptors so that the array of G protein couples receptors to distinct intracellular targets. For example, G protein associated with the epinephrine receptor is called GS. Okay, this is like stimulatory. GS stands for stimulatory. So it is going to activate your adenylyl cyclase. Okay, these are important, guys. These are important points. Other G protein, alpha and beta gamma subunits act instead to inhibit adenylyl cyclase or to regulate the activities of other target enzymes. In addition, both alpha and beta gamma subunits of some G proteins directly regulate ion channels. So what we are saying is that if it is GS, it is stimulatory, your alpha subunit gets activated. Okay. If it is inhibitory, it's not alpha, but it is beta and gamma. Okay. So obviously there, it is not going to affect the adenylyl cyclase, but the other proteins. Clear? Now, let's see how this GPCR is getting, how the G protein is getting regulated. Okay. So first thing first, so we have got the GPCR here. Okay. So let's start from here. So this is out, this GPCR is in the inactive state. Okay. So what is happening in the inactive state? The alpha is bound to your beta gamma. Correct? This is the first step. Now, with the help of those regulatory proteins, what is happening then? The GDP is being replaced by your GTP. Okay. This much is clear, right? So hormone binding, which will result in the stimulation and the release of GDP and in exchange of GTP. So hormone goes and binds, conformational changes, your alpha is active now. Correct? Now, so this gets dissociated from the beta gamma. This alpha is active. Now it is going to target the enzymes for the ion channel depending on the signal which it has received with the help of these hormones. Okay, so let's say that it goes and activates your adenylyl cyclase and it results in the increased concentration of your CAMP, cyclic AMP. Okay, now this activated GTP bounds alpha subunit and beta complex then dissociate. This is the same thing. They dissociate and this gets activated. Now, so activity of alpha subunit is terminated by the hydrolysis of the GTP. So, जैसे GTP hydrolysis होगा, it will become GDP, right? And this hydrolysis is maintained by the RGS. What is RGS? Regulator of your G protein, right? So then the inactive GDP bound alpha subunit will again reassociate with the beta gamma. So that's how your G protein is getting regulated. Okay, simple, right? It's clear. अब यहाँ से थोड़ा ध्यान देना शुरू करना, okay? I mean, of course, not thoda, I need more concentration. So how the secondary messengers and the phosphorylation is actually, resulting in the signaling. Sorry for that, slide is not made properly. So the role of CMP, it is a major target of your G protein signaling in mammalian cells. For example, the it stimulates the breakdown of glycogen to glucose in muscle cells. So what is the first thing over here? Increase in CAMP means more glucose is required. So what is happening? Glycogenolysis is happening. So the action of epinephrine is mediated by increase in the intracellular concentration of CMP, leading to the concept that CMP is the secondary messenger in the hormonal signal. Okay. Now it is formed from ATP by the action of adenyl cyclase, which is the stimulation over here. So ATP से आपका cyclic AMP बना. Okay. Now if suppose I need only AMP, so what is happening now? So adenyl cyclase, cyclic AMP. CMP phosphodiesterase, what it is doing? It is breaking the cycle. Can you see this cyclic AMP हम क्यों बोलते हैं? Because it is bound in both the sugar, right? At 3 carbon at the 5 carbon dash. So that cycle is broken by the phosphodiesterase, and what you get is only the AMP. Clear? This is clear, this part?
Yes.
Okay. So most effects of CMP in animal cells are mediated by the action of CMP dependent protein kinase, for example, protein kinase A. So this also we have done in the previous lecture. In the mole bio, I think we have done like this. Okay, so it has got two catalytic subunits and two regulatory subunits are there. Okay, so cyclic AMP will go and bind to the regulatory subunits here, like this, leading to their dissociation from the catalytic subunit. So what will happen? These two are getting free. Okay, then now this will go and get phosphorylated. Okay, so the free catalytic subunits are then enzymatically active and able to phosphorylate serine residues on their target proteins. So they are protein kinase A, यानी कि phosphate group add करेंगे. When they can add? When cyclic AMP goes and binds to the regulatory subunits and these two CC, okay, they get activated. Okay, now they can add phosphate group on the target proteins. Now, in the regulation of glycogen breakdown by the epinephrine, the key target of protein kinase A is another protein kinase called phosphorylase kinase, which is phosphorylated and activated by protein kinase A. Okay, we'll come to this. So phosphorylase kinase in turn phosphorylates and activates glycogen phosphorylase, which catalyzes the breakdown of glycogen to glucose 1-phosphate. I hope guys you're not getting confused here. So सबसे पहले क्या है? Let's talk about phosphorylase kinase. So what is happening? You have got the glycogen. Okay, as I said, I need more glucose. So glycogen to glucose 1-phosphate. Okay, this is done by your phosphorylase kinase. ठीक है, ये kinase इसके एक molecule को break करके glucose 1-phosphate दे रहा है. Right? Is this much clear? Now, who is activating phosphorylase kinase? Protein kinase A is activating. When protein kinase A adds phosphate group to the phosphorylase kinase phosphorylase kinase, it then goes and breaks down your glycogen to glucose 1-phosphate. Is this much clear?
Yes.
Okay. So what is happening? So this is how the, I'll show you the cascade first, how this is being done. So you get a signal. Okay. For example, epinephrine is there. So epinephrine goes and binds to your GPCR. Okay. It goes and binds to your GPCR. GPCR से यहाँ पे आपका अल्फा सबयूनिट एक्टिवेट हो गया. अल्फा सबयूनिट आपका AC एक्टिवेट कर रहा है. इससे क्या होगा? आपका cyclic AMP इंक्रीज होगा, right? Cyclic AMP will go and bind to your regulatory subunit of your protein kinase A. CC यहाँ पे आपका activated हो गया, catalytic subunit. Now this protein kinase A will phosphorylate your phosphorylase kinase enzyme. Okay. Now this activated phosphorylase kinase will go and break down your glycogen to your glucose 1-phosphate. Okay. So each molecule of epinephrine activates only a single receptor. So each receptor, however, may activate up to 100 molecules of GS. And each GS can stimulate enzymatic activity of adenylyl cyclase, which can catalyze the synthesis of many molecules of CMP. So like this, what is happening then? So this signal is getting amplified. Okay, so signal amplification continues as each molecule of protein kinase A phosphorylates many molecules of phosphorylase kinase, which in turn phosphorylate many molecules of glycogen phosphorylase. Okay, so resulting in what? We are getting so many molecules of glucose 1-phosphate. This is the same thing which I was just explaining now. So epinephrine goes and comes and binds to the GPCR. Alpha protein is getting activated. So here GDP jagah GTP aa jayega aapka. This goes and activates your adenylyl cyclase, which results in the conversion of ATP to CAMP. CAMP will go and bind to the regulatory subunit of your protein kinase A. Catalytic subunit is now free, which will add. Okay, it's a kinase. Catalytic subunit is a kinase. Goes and adds the phosphate group to your phosphorylase kinase, makes it activated. Now this activated phosphorylase kinase will activate your glycogen phosphorylase, which will add, which will convert your glycogen to glucose 1-phosphate. Okay, and now this can go for your glucose or glycolysis. Clear? Is this figure is clear to everyone? Tanvi, Krishita, Akshaya?
Yes.
Yeah, it is important. Okay, please remember this, because they might ask, like, agar, if I have inhibitor at this step, then what will happen to the rest of the downstream step or the upstream? Agar suppose let us say that I added the inhibitor of your adenylyl cyclase, so what will happen? So there won't be any generation of CMP, so no glycogen breakdown will happen. Okay, so like this questions can come. Now, in addition to this, hormone binding to a small number of receptors leads to activation of much larger number of intracellular target enzymes. Okay, so in many animal cells, increases in CMP activate the transcription of specific target genes that contain regulatory sequence called CRE, CMP response element. So in this case, the signal is carried from the cytoplasm to the nucleus by the catalytic subunit of protein kinase A, which is able to enter the nucleus following its release from the regulatory subunit. So just now we saw that the catalytic subunit is getting free from the regulatory subunit when the CMP is bound to the regulatory unit, right? Now this catalytic subunit will go into the nucleus. Within the nucleus, protein kinase A phosphorylase transcription factor called CREB. CREB is for CRE binding protein, leading to the recruitments of co-activators and transcription of CAMP inducible genes. Now, such regulation of gene expression by CMP plays an important role in controlling the proliferation, survival, differentiation of a wide variety of animal cells, as well as being implicated in learning and the memory. Okay, so ऐसा नहीं है कि सिर्फ glycogen breakdown हुआ है, it also goes at the nucleus level and activating it. So cyclic AMP inducible gene expression. So ये common रहा आपका, ठीक है? That hormones comes and binds, G protein getting activated, activating adenylate cyclase, CMP goes and binds the regulatory protein. Now C is also activated, right? It is free from this. Now it goes into the nucleus, where it is going and phosphorylating the CREB. What is CREB? CRE binding protein. Okay. Now, of course, it's a protein kinase A. So, ये क्या करेगा? Phosphorylate करेगा CREB को. So now they go, the CREB, it gets activated, goes and binds to your CRE binding sequences here. It's a binding protein. So ये यहाँ bind करेगा. It also goes and the core activators comes and binds to the CMP inducible genes. So together they are resulting in the transcription of the genes, which is responsible for the cell survival or differentiation, etc., etc. Clear? So how one hormone can affect the pathways? See, of course, for the survival, of course, for the proliferation, you need glucose, right? And then only the cell can divide. So this is how it is helping in the transcription of the cyclic AMP gene expression. Guys, this is clear? This figure. So it is like half of the CA is responsible for the nucleus gene expression and half of them are responsible for the glycogen breakdown. Any doubts here, Krishita? Tanvi, is it clear? Okay. So now it is important to recognize that protein kinase such as protein kinase A do not function in isolation within the cell. So we have got the protein kinase A. So target protein activate karega by giving the phosphate group over here. But if suppose I don't want this any other, further signaling, then I need phosphatase. So protein phosphatase 1 will come and remove this phosphate group and make it as the inactive. So this will keep on going till the signal is being terminated. Okay. Yeah, that's all guys. This is all about the GPCR related signaling. Is it clear to you like how GPCR is involved in the signaling? Okay. So yeah, so I'll be stopping here now. 1 second.